Hello, Forensic Science. This video is our lecture 3.2. It's going to be talking about testing red blood cells and blood typing and something called the Kastle-Meyer test. So, there are all kinds of different substances out there that could be mistaken for blood, right? There's ketchup, paint, barbecue sauce, makeup blood or like fake blood, things like that. So, scientists devised a way to quickly and efficiently determine is it blood or is it not blood? So, the test that's used to determine whether it's blood or not is called the Kastle-Meyer test. The Kastle-Meyer test is a type of a particular kind of what's called a presumptive test. We're going to see presumptive tests a lot for the remainder of the school year. Essentially what a presumptive test does is a few things. One is it tells you a simple yes or no. It's always testing is this thing present or is it not present? But it doesn't tell you anything additional. It only tells you, is this thing present or is it not present? A COVID antigen test is sort of like a presumptive test. It simply says, does the person have COVID or do they not? It doesn't say whether you have a cold. It doesn't say whether you have the flu or another virus, right? It doesn't tell you the other information. It just tells you, it just tests for that one thing. And that's how presumptive tests work. They just test for the one thing. And they're done because they're usually cheaper than the more complicated tests, like for example, for COVID that would be like a full-on PCR test, for example. And they're actually faster typically, right? Your antigen test at home is very quick. It's only 15 minutes kind of thing, whereas the PCR tests could take you a couple of days to get results from. So we do the Kastle-Meyer test first on the blood. It's one of the very first things that's done because we don't want to have to go through and do all this other testing that's more complicated and more expensive right away. So if it would be silly, for example, if we had some ketchup and we took that ketchup and we were like, all right, let me see if there's DNA in it. Let me go through and do this whole expensive testing set. And it's like, oh, it's ketchup kind of thing. Same thing with this. So the first thing we're going to do is apply the Kastle-Meyer test and see, is it blood? And if it is, then we can go from there, right? Then we can test further, try to figure out what blood type, and also try to figure out if there's any DNA in there. Maybe we can actually nail this down to a specific person kind of thing. But the overall result of the test is relatively simple. If blood is there, it will turn pink. And if blood is not there, it will stay the same color that it already was. So here's another visual of that. So you take your unknown dry red substance, you don't know if it's blood or not at this point, and use a cotton swab and rub some of that substance. Then you apply the Kastle-Meyer test. So you add a drop of phenolphthalein. You may remember phenolphthalein from chemistry. It's an indicator for acids or bases. So you put the phenolphthalein onto the blood sample, and then you add some peroxide as well, and the blood combined with hydrogen peroxide and phenolphthalein will then have potentially a reaction to show pink or to not show anything if the blood's not there. If the blood, so again, this is a negative result over here. Notice how it's still red. The color of the blood was at the beginning. And a positive result is pink. So if it turns pink, there's blood. That's the summary of the Kastle-Meyer test. So, human blood types, though. A little bit of a review from biology. I think the best way to think about this is to think of, just like in our picture here, people's red blood cells having particular flags on them. Technically, those flags are referred to as antigens. So, for example, a person who is type A blood will have A flags on their red blood cells, or in other words, A antigens. A person who is type B will have B flags on their red blood cells, or in other words, B antigens. So then if we look at it that way, it makes sense that someone who is type B cannot donate blood to someone who is type A, because that type B coming into type A will see an unfamiliar flag. It will see the B flags and say, whoa, 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 we don't recognize that in our body, and the person's body will have an immune response where it will reject that blood coming in. It will say, no, it'll actually attack the blood as it comes in because it sees it as a foreign entity. It doesn't recognize those B flags. The reverse would happen too, right? Someone who's type B blood would not recognize type A's antigens, type A's flags. And so it would see it as a foreign invader coming in. But if we look at type AB, for example, type AB has all the different kinds of flags. It's got type A and type B's flags. So it will welcome type B. It will recognize its flags, its antigens. It will say, come on in. Same thing, it will also accept type A blood because it will recognize those flags, those antigens. It'll say, oh, I know you. I already have you on me, kind of thing. And it will accept that. But the reverse would not happen, right? If type AB tried to donate to type B, then the A portion of AB would not be recognized and it would be attacked. If type AB tried to donate to type A, then the B portion of AB would not be recognized and it would be attacked. So, with that being said, type AB is considered to be the universal acceptor, if you remember. It can take any kind of blood because it recognizes type A's flags and type B's flags. And if we look at type O actually has no antigens or no flags. There's a rumor that someone actually meant to write down type 0 blood, as in 0 antigens, and then someone would misread it as type O, and we've gone with that ever since. I don't know if that's actually true, but it helps to remember that, okay, type O actually has no antigens on it. Type O cannot accept blood from any of these other blood types because everything would be foreign to it. It doesn't have any of its own flags or antigens to recognize anything else coming in, so it would attack anything coming in. But because type O has none of its own flags, it is the universal donor, right? All of these other ones could take blood from type O people and nothing would trigger in the body. There would be no flag for the body to say, hey, that's a foreign invader kind of thing. It would just accept it as its own blood. There'd be no reaction there. So, that's some of the basics of blood types. We're going to go into more in a second, but before we move on, I just wanted to say, blood is class evidence, right? Blood type, I should say, particularly is class evidence. Multiple people could have the same blood type, right? It doesn't tell you enough information. Blood itself is going to be individual evidence if it has something that we'll talk about another time. But for now, we want to know blood type, as in AB, O, and AB. That's going to be class evidence. Knowing the type alone will not be enough to nail it down to a single individual. But what about the positive-negative part? Okay. The nice thing is that the positive-negative part simply means one more antigen, an antigen that we call the Rh factor. If someone is Rh negative, they do not have the Rh antigen as a cell surface protein. They don't have it on their red blood cells. If they're Rh positive, they do have the Rh antigen. So, again, if we think about COVID, right? If someone's positive, that means they have COVID. If someone is blood type positive, that means they have this Rh factor. Positive means, yes, you've got it, right, in medical testing. Same thing here. If someone's negative blood type, that means they don't have that Rh factor. So it's just one more flag, one more thing to keep track of. So then, because of that, this person is actually blood type A positive, right? Because they have A and the Rh factor. This person's A negative because they just have A and they do not have the Rh factor. They're negative for it. They don't have it. Okay, so then here's our actual 8 blood types. Before we looked at only 4 of them, but realistically we know there are 8, right? You could be AB positive, A positive, B positive, O positive, or AB negative, A negative, B negative, O negative. Here the black squares are the Rh factor. So all the top ones have the Rh factor, so they're all positives. All the bottom ones do not have the Rh factor, so they're all negatives. So then we kind of need to revise who is the universal recipient of all blood types, right? More particularly, it's not just AB, but it's actually AB positive who is the universal recipient. If someone is AB positive, they will be familiar with the A antigens, right? The yellow flags here. The B antigens, which are the blue flags here. And the Rh antigens, the Rh factor, the black squares. So any blood that's coming in for that person will not be unfamiliar. They have all the flags. They're like a country that accepts everybody kind of thing. So nothing will be unfamiliar for that blood type. If we look at the other extreme, though, O negative, right, would not be able to accept blood from anyone but O negative. Because even if someone was O positive, O negative would react to their Rh factor antigen. It would react that way. There would be an immune response. So O negative can only accept O negative. But what's nice, though, is that if someone's O negative, they can donate to anybody, right? They legitimately have no antigens on their surface that would be recognized for an immune response. So every other blood type on here would be able to accept this blood type because of that. So AB positive is the universal recipient. O negative is the universal donor, can give to anybody. AB positive can receive from anybody without having that reaction of that rejecting the blood coming in. All right, so let's take a look at that. These Y shapes are what antibodies actually look like. They're like a Y shape, and the binding site is at like the top left and right of the Y shape. We don't need to go too much into it. It's just that we need to know that if the blood types are not compatible, then these antibodies will actually come out and attack the incoming blood, right? If it's not one of those compatible blood type relationships between donor and recipient, then the recipient's antibodies will attack. They'll treat it as a foreign invader, and what will happen is they'll cause something called agglutination to happen. Agglutination is a fancy term for clumping. So it'll take those red blood cells and actually cause them to clump together and be rejected. So here's that happening. Here's the antibody without it being bound to anything. Here are antibodies binding to red blood cells that they see as foreign invaders. And here is the blood clumping or agglutinating together in agglutination, causing that clumping to happen. So they have to be compatible, or the person receiving the blood transfusion will die, right? This clotting will happen in their blood, and they will not survive the interaction. So they need to be compatible. You can't just give any blood to anybody kind of thing. Okay. I think so far it's kind of made a lot of sense, right? But this is the part you kind of have to scratch your head and think a little bit harder about. Blood type A, if someone is blood type A, right? They make antibody B. So if they're blood type A, remember the only flags that they recognize are the flags of A. They produce antibodies that go and hunt out and find blood type B if it should come in. Right? So they make antibodies, they make soldiers that hunt out the enemy kind of thing. Now if someone's blood type B, they make antibodies against blood type A. So that makes sense if you really think about it, right? Like their soldiers are hunting for other kinds of foreign invaders like blood type A. If someone is, it should say if they're Rh negative, then they make the antibodies for Rh. So if someone doesn't have the Rh flag in their system, then they actually make the Rh antibodies. They make the antibodies that go and hunt that down. So here's kind of a visual of that. I think this will make more sense. If I'm blood type A, I produce antibodies that are looking for B. Because the antigens that I have are A. I have A flags on me. If I'm blood type B, I'm producing antibodies that look for blood type A. Because I have B flags. If I'm blood type AB, I have A flags and B flags. Those are my antigens. So I don't make antibodies that look for A or B because then I would literally be attacking myself. If I'm blood type O, then I make antibodies that try to hunt down and stop both A and B. Because both of those are foreign to me. And I don't have any antigens on my surface. Okay, so that takes a little bit of processing. You may need to watch that again for that to make sense. But again, the last one is that if someone doesn't have Rh factor, then their blood looks for, it has antibodies against Rh. Okay, but so as far as how to actually perform the test, what we do is we get a tray like the one shown at the bottom right. And there's going to be 3 wells in that tray. You're going to be doing this in class. 3 wells in the tray. There's an A well, a B well, and an Rh well. This is the part that gets a little confusing, so you have to pay extra close attention. in each of those 3 wells, you're going to put the same blood source. So let's say it was unknown blood from the crime scene. We're going to put crime blood there, and crime blood there. All 3 of them, they're going to have the same blood put in. What's going to be different, though, is that in well A, you're going to add antibodies that are against type A, antibodies that look for type A. You're going to add those to well A. In well B, you're going to add antibodies that look for type B antigens. And in the RH well, you're going to add antibodies that look for Rh positive. So, if those antigens are found by the antibodies, then the blood will actually clump up and it will look like what's in B well over here. If it's not found, then it will continue to look the same as it did. So the way we interpret these results is that we didn't see any of that agglutination, that clumping over here, so that means A was not present. This person's blood type does not contain A. There was no clotting over here either for the Rh, so that means that the person's blood type does not have Rh either. In other words, they're negative. And then finally, there was some clotting in B, so that means that, oh, okay, this person has B-type blood, but they're B negative because there was no Rh. So the summary is where the clotting happens is the type that they have. And where there's no clotting, it means they don't have those things. This person didn't have A or positive. They just had B. And this testing is done because it's less expensive than DNA profiling. To actually go through and have to do a DNA analysis to find this out is actually much more helpful to do blood typing and to decide if you want to investigate further. Does the blood type match, or can it help the case at all? But okay, so here's other examples. Oh, actually, here's our initial setup, and this is what we were just talking about, right? This is the same exact thing we just saw. Initially, it'll just be the blood, and then in this case, then we add the antibodies, A to A well, anti-Rh antibodies to Rh, and anti-B antibodies to the B well. And then there was no A present, no Rh factor present, but B was present. So the person is B negative, in other words, because they had B and they didn't have the Rh factor. Our last 2 examples here. So 2 blood samples were found at the crime scene. The antibody antigen blood typing test was performed. Analyze the results below and determine the type of blood for each sample. The regular red circle means there was no clotting, no agglutination, and the red and white means there was clumping. So think about what this means. There was no clumping for A. Right? When antibody A was placed in the presence of this blood, it found nothing to attack. When antibody B was placed in the presence of the same blood, it found nothing to attack. But when anti-Rh, the antibody against Rh factor was put in, it found something to attack. It agglutinated. So then this blood type would be O. This is the hard part. Would be O, right? Because it doesn't have A or B. A or B was not found. There's no clumping there. So this would be type O. And particularly this would be type O positive because the Rh factor was found. There was clumping happening there. Agglutination happened. For this last test over here, there was detected antigen A. Antibody A found something to attack. So there was clumping. Antibody B also found something to attack. So there was clumping agglutination here too. Antibody Rh, who looks for Rh, didn't find any. There was no clotting. So then this person's blood is actually AB negative because there was no Rh factor. So that's it for our blood typing. We're going to practice more in class. Thanks for watching, and I'll see you soon.